Hi, in this video I want to explain how polymerase chain reaction works, and we start this reaction with uh, single-stranded DNA or double-stranded DNA. For my example I will use single-stranded DNA, and we design primers that going to anneal to one strand of the DNA, and for the second strand of the DNA, no matter how many strands of the DNA we have for the beginning, our reaction would include both strands of the DNA. So we have to design our primers that would anneal to the one end of the strand of the DNA. So we have two kinds of the primers, each for each strand of the DNA. And for example, this one would anneal to this end, and here is going to be 5' end, and here is going to be 3' end, and a new strand of the DNA always extend from the 5' end to 3' end. And two strands are reverse complementary to each other, so this is going to be 3' end here and 5' end for this strand. So also with the help of the Taq polymerase, this is abbreviation of the T. aquaticus bacteria, and this enzyme were derived from this bacteria that lives in hot springs and can withstand hot temperatures, and we need such enzyme here because we have cycles when we have to rise temperature and then lower temperature. We have to rise the temperature in order for the two strands of the DNA to melt or dissociate, and then we need to lower the temperature, so our primers that we designed for both strands of the DNA would be able to anneal, and Taq polymerase would be able to extend a new strand of the DNA. So when temperature lowered, our polymerase chain reaction starts, and polymerase extends this DNA strand until it hits trinucleotides that code for stop, or we also call it stop codon, or until they are going to be the end of the fragment of the DNA. So this is our first reaction, so we can call it first step. And with the second step we have two strands of the DNA, initial DNA, first strand of the DNA, and now we have second strand of the DNA, and we rise the temperature, and these two strands dissociate, but not the way how, for example, ice melts, because here just hydrogen bonds would break, and when temperature would be lowered, these two strands of the DNA once again would form a double-stranded molecule. So this is not the change in the aggregate state, and once again we have here our primer. We can call it primer number 1, and this is going to be, for example, primer number 2. So our primer number 1 would once again anneal to the initial strand of the DNA that we start with, and primer number 2 this time would anneal to the other end of the new strand of the DNA that we got after the first cycle. So this is going to be second step, and with this step once again polymerase would extend new strand in this direction, so this is going to be 5' end and 3' end, and 5' end here, and 3' end here. And this strand of the DNA also would be extended in this direction. So we started with with single-stranded DNA, and at the second cycle we already have 4 strands of the DNA. So with each cycle we have more and more product. This is exponential growth. So it's counted like 2, 4, 8, 16, 32, and so on. We also might get our initial product. For example, our strands that we started with might be very long, and we need only product that can be, for example, of this length. We just need a fragment, so we just design our primers that would make only this particular fragment that we're interested with. And of course, every time with each cycle we still have this strand of the DNA present, and this strand of the DNA present if we started with double-stranded DNA, and we also will have half products. What does it mean? For example, if we extend this strand in this direction, this is going to be of the same length as this strand of the DNA, or a little bit shorter. It starts from our primer. So it's going. This particular strand is going to be half length, and this one would be full length. So this is going to be full length, and this is half. And we also will get a product with the next cycle when we have 2 half products, to which 2 primers that we designed would anneal, and will produce a product of the desirable length. So these 2 strands of the DNA that we start with, that is full length, that these 2 would be present through the all the cycles, and these half length products would grow in arithmetically. So with each cycle we will have we will count them like 1, 2, 3, 4, 5, and so on, and our product of the fragments that we looking for would grow exponentially, like 2, 4, 8, 16, 32, 34, and so on, 64, and so on. So we after 20 or 30 cycles, product that we are interested in would be present in millions or billions copies, and these products would be just negligible and even won't produce background noise. But background noise can be produced if we designed these primers not referring to the rules of how to design primers. For example, such a rule that two primers shouldn't be, their sequence shouldn't be anywhere complementary to each other, or these two primers may form dimer primer, and our PCR reaction would stop. And also another rule that primer, we can call this primer 1 or this primer 2. Also, if we are making a copy of the gene, then in this case we can call one is forward primer and another one is reverse primer. Also we can call plus and minus, so we can give many names, it's up to you how to call them. The second rule would be to design these primers in such way that the melting temperature would be the same. And also these primers should be, the length of this primer should be about 20-30 nucleotides, and this give us enough uniqueness, so such a primer would anneal to a particular place on the DNA that we are interested in. So 20-30 nucleotides length would be enough, for example, for such primer to anneal to only one place in the whole human genome, because 30 nucleotides would be so unique that it's going to exceed the whole number of the nucleotides in human genome by number 2 or 3 times. So another rule would be not to use for the primer such fragments on the DNA that is AT rich, or for example have many repetitions of the same nucleotide sequence, for example TTTT, because in this case such primers on the first place lose their uniqueness and also may breathe when forming loops and any other undesirable structures. So there are many different rules. I won't explain all those rules in this video. I have about 10 more videos where I'm explaining those rules, how to design primers. And probably the last thing that I want to mention today is that those we have polymerase chain reaction that is show us exponential growth, but still this reaction would you would see effect of plateau when after 20 or 30 cycles you won't get more product, but background noise would grow with side products that would contaminate your sample. So we usually don't go over the 30 thermocycles, and this is between 20 and 30 thermocycles when you get the best results. And this is all for today. Thank you for your attention. Goodbye.